You'll also need to work on mammalian cells in your lab exercise. So let's talk about mammalian cell cultures, different strategies, different types of cultures and different strategies you use to culture mammalian cells. So first of all, why do we need to culture cells? There are several reasons. One of them is clinical, other is of course for usage in science and research. In vitro cultivation of cells from different tissues is basically referred to as the cell culture. Vertebrate, for example, can have more than 100 different cell types. Each cell type generally requires specific culturing conditions. So as we were talking about why we need to culture cells in vitro, because one of the things, beauty about culturing cells is that you can manipulate the environment of the cells. So you can see how different cells will behave in a specific environment. You can change pH, you can add different reagents, you can add different signaling molecules that we have talked about in theory and see how cells respond to them. So clinically, when you're using cell culturing, it is very important. People use for clinical usages, for example, fertility treatment. You know about test tube babies, quote unquote, how they're made. That is done through cell culturing, of course. Chromosomal studies, when we want to look at chromosomes of a person for whatever reason, we need to culture cells. And when we culture cells, we have to get them at a division phase, for example, metaphase, where the chromosomes are maximally condensed, so we can look at chromosomal aberrations. So we need to culture white blood cells, for example, or amniotic cells to look at the chromosomes. We have talked about tissue engineering and also organ engineering. If you need to do those things, you also need to culture cells. Additionally, people use cell cultures to test out new drugs, for example. If there's a drug and they want to do a study to ascertain toxicity of that new drug, for example. Of course, traditionally, people can do that on different animals, rats or mice or rabbits, for example. But these animals are not human beings. Human tissue has the potential to respond differently to a particular agent than these animals. So we can expose human cells to these different drugs which we want to test, for example. In research, as I've mentioned, to study cell structure and function, we need to culture cells. Also study behavior of the cells, and also look at study the developmental stages and responses of cells to different stimuli. Generating monoclonal antibodies, we didn't talk about that when we were talking about immunology. It's a different subject. However, I'm sure you have come across the term monoclonal antibody. So you need to culture cells to generate these antibodies, and they play a very important role for different types of diseases. Generation of recombinant proteins. For example, we have talked about how we can ligate two different pieces of DNA, and once that DNA is introduced into cells, the cells start producing a protein corresponding to that particular DNA. In order to do that, we still need to do cell culturing. If we want to study viruses, for example, we know viruses cannot exist independently. They have to live inside the host cells, which you won't be able to generate host cells unless you do cell culturing, for example. So now let's talk about types of cell cultures. The primary cells are cells straight from the tissue. Those are the primary cultures. There's no passage. Passage basically is a technique that once you have grown cells, we will talk about passages in more detail later on. But anyways, for now, I would just like to mention passage is when you're growing cells in a container and the container gets full. So you remove these cells and you split these cells into, for example, 2 or 4 groups, and you can put them in a new container. So that is basically passaging. Primary cell culture may compose of mixture of cells. So if you're taking a tissue from an organism, it can have different types of tissues. Fibroblasts can be present pretty much in most of the tissues. Cell lines have at least one passage. You will most likely be dealing with cell lines in your lab. With each subsequent subculture, there are several means of obtaining immortalized cell lines. There are different types of cell lines that the cells from these cell lines can grow, divide and multiply indefinitely. Because you've talked about cancer cells when we were talking about oncogenes or neoplastic transformations, cells have a clock in them. They can count the number of times they have divided. Generally, most cells divide about 50 times, and after that, they are not capable of dividing or they cannot function normally. So, however, cancer cells or tumor cells, they can divide indefinitely. So, those are the immortalized cell lines that people use in their lab for different purposes. Pure cultures are basically cultures of cells that have a single cell type. So how do we obtain pure cultures? There are different ways. One of the ways is that you use reagents that will promote the growth of the specific cell type that you are interested in, and it will prevent other cells from growing. You can also use different techniques. For example, we'll talk about it later on. Magnetic sorting, cell sorting. I'll tell you about that technique later on. But you can use different techniques to separate cells, a specific type of cells. Grossly, we can divide cell cultures into two groups: adherent cell culture or suspension cell culture. Adherent cell culture is appropriate for most cell types, including primary cultures. Suspension cultures, these are the cells that do not stick to a substrate or the bottom of the dish. They stay suspended in the media. These are generally the cells we culture through suspension cell culture method, are the blood cells or the bone marrow cells. Adherent cells require periodic passaging but allows easy visualization and inspection under an inverted microscope. The suspension cells, of course, you have to passage them also, but the way you passage them is slightly different than the passaging of adherent cells. And also, since they're suspended, it will be hard to observe these cells under an inverted microscope. Adherent cells have to be dissociated enzymatically or mechanically. We'll talk about that later. However, suspension cells, they are generally, since these type of cells, that they don't stick to each other or they don't stick to the extracellular matrix molecules, so you don't have to dissociate them enzymatically or mechanically because they're already single cells. Explant cultures, a piece of a tissue is placed in an appropriately coated dish which contains the media. Cells typically move out of the tissue and start to proliferate in the dish. This is useful for sensitive tissue which may be prone to damage by protease or enzymes that you need to make tissue fall apart, so cells will separate from each other and they will adhere to the culture dish. Also, in case of neuronal tissue, explants provide an excellent opportunity to the scientists to have a dense area of axons with a uniform polarity of their microtubules, for example. So this dense area, if someone is interested in studying axons, they'll place a piece of neuronal tissue, cell body mass, for example, CBM, or if it is a neuronal tissue, the cells will extend axons out. If it is a non-neuronal tissue, cells will generally crawl out of the explant and they will start covering the dish. So let's talk about the dissociated cultures. Tissue sample is dissociated either mechanically through trituration or enzymatically, generally a combination of both, producing a cell suspension. So what is trituration? You take a glass pipette, you flame it. So when you flame the glass pipette, the pore or the hole of the pipette becomes smaller. Here, for example, we are flaming the tip of the pipette. It was 1 millimeter approximately initially. After flaming, it became approximately 0.2 millimeter. So when you triturate a tissue, basically when you apply negative pressure, the sample will go inside this pipette and then you blast it out. You do it several times till the tissue dissociates. This is how you mechanically dissociate tissues, for example, to obtain individual cells. Cells are plated on coated dishes, culture dishes, and of course, these cells will settle down and they will start to divide. Next day, you have to come in, of course, change the media. That will do two things. First of all, the cells will get fresh nutrients. And also, when you change the media, dead cells and any toxic materials that were released by these cells when they were dying, they will also be removed when we change the media of these cells. So we have seen some techniques of culturing mammalian cells. We'll continue our discussion with this topic.